I'm a microbial ecologist. I'm here at the Ecology and Evolution Department and also Department of Surgery. And I have a fundamental interest in bacteria and how bacteria assemble in myriad environments. Literally, there are bacteria everywhere on Earth. Then there are lots of bacteria inside you, but they're also inside the oceans, and they're under the crust of the Earth, and they're in the air, and they found every niche that's possible to actually become part of, they're found there. So they are very, very important. Today, I'm really going to focus in on some of the work we're doing in the human microbiome, which, as you're eating lunch right now, is going to be highly relevant to you guys. And so hopefully that will help us to understand a little bit about what's going on inside your stomachs right now. Right, so you're a microbial organism. About 3 pounds, 2 to 3 pounds of your body mass is microbes. That's bacteria, archaea, viruses, and fungi. It's about the same weight as your brain. And as a lot of my colleagues like to say, this is an organ. It's another organ, a part of our body, which is fundamental to our health. And we're only really just starting to understand how fundamental it is. Really only in the last 10 years or so that we've really started to take notice of the microbes in our body as an effector of our neurological, our psychological, also our physiological, and even our hormone levels. Our well-being in general is predicated on what's happening to these little guys inside us, right? And what happens to them is very, very important at certain different levels. If we keep them happy, we may be kept happy. And that's what we're mostly trying to focus on. So we find them, if you think of the human body as just another Earth, there are loads of little niches inside us. There's, moist, wet niches, and then there's anaerobic niches inside our stomach where there's no oxygen. There's cold, dry areas, like on our elbows or on the back of our hands. And all of those select for different types of bacteria and archaea and microbial organisms that want to live there. We need microbes. They're essential to our body. If we grow a mouse up without any bacteria inside its body, any viruses, without any fungi, it becomes a little bit distorted, right? These are mouse stomachs. So the one on the far right is a healthy mouse stomach, the little one, okay? Mouse intestine. The one on the far left is a mouse intestine that's grown up inside a mouse without any bacteria inside it. And it's inflated, it's distorted, right? Because these animals have evolved in the presence of constant bombardment by microorganisms. So it's not like they've evolved in isolation. Evolution never occurs in isolation. It occurs in an environmental context. The environmental context of mammals and all living animals has been bacteria and archaea and fungi, right? So it's evolved an immune system to cope with this constant exposure. And as I'll show you a bit later on, it's actually evolved an immune system to actually actively grab some of those organisms and use them for its benefit, right? But in this system, we take them away, and the immune system's overreacted to the absence of microorganisms. The absence of microorganisms has caused the body to pervert the development of certain organs in the body. And we also know it perverts the neurological behavior, right? In this experiment, you've got an elevated maze. It's not a very complex maze, it's just a crisscross. But it's got a little isolated box on either side. If we stick a normal mouse in that box, you'd expect the mouse to hide away and hunker down, because a good evolutionary response for a mouse is to not be spotted by a predator, right? So it stays where it's dark, where it can't be seen. If we take all the bacteria away from that mouse, it runs outside of the box along the platform and jumps off the edge of the platform and goes crazy, okay? Its neurological behavior has been perverted by the absence of microbes. And we can add in bacteria back into that mouse, different types of bacteria, and alter its anxiety, its response to its environmental stimulus. This is really important. The way it does this is by affecting our immune system in our gut. Different bacteria and different bacterial communities inside the intestine of this mouse are interacting with the immune system. They're releasing things called cytokines, little chemicals. And these cytokines interact with our neurological system, sending nerve impulses up the vagus nerve, through the spine, up into the brain, and releasing certain facsimile-like compounds, different types of cytokines, which bind onto our neuroreceptors and change our behavior. This happens while you're eating right now. Your microorganisms are actually responding metabolically to the contents inside your gut, and that's changing your microbial community composition over the next hour, and it's actually going to start to affect neurological behavior in you. So, this can be things like just your appetite starts to fluctuate, or it can be other things like mood swings or anxiety. And if we can harness this, and if we can understand the metabolism of these organisms at their core basic level and how they respond to those stimuli, we can come up with real translational impacts to affect things like depression and anxiety in the human population. Very important. We know from work with several of our colleagues, Diamantinopoulos at Argonne National Laboratory and Kathy Nagler here, sorry, that we can change the response phenotype of a mouse that's severely allergic to peanuts. If we add in certain bacteria back into this mouse, certain clostridial bacteria, which produce a compound called butyrate, then that mouse will stop responding negatively. It will stop overreacting to that stimulus of peanuts, right? So we can actually cure a peanut allergy in a mouse. And what Kathy hopes to do with several of us and around Chicago is to develop an initiative, a center to really help us to take in money and produce real translational impact upon patients who have food allergies. So a food allergy prevention initiative. And this is all predicated on our understanding of how that system works. We know some bacteria can make you fat. This is really important. We have an obesity epidemic. And if we consider what an epidemic is often a disease condition, right? And we actually think that in some instances, our lifestyle has affected a change in our microbiome, which is eliciting this disease phenotype, right? In this instance, the obesity. All right, so how does that work? Well, let's look at what the causative factors are. This study by Liping Zhao from Shanghai from 2012. Liping used to be a very, very heavy guy, right? He weighed 385 pounds. He took a diet for 9 months of traditional Chinese wheat, whole grain food, and he lost 113 pounds in about 6 weeks. Sorry, 6 months. 6 weeks would have been crazy. That's my own story. I'll tell that in a minute. But in 6 months, he lost 113 pounds, which is still pretty crazy, right? And he checked his microbiome at the beginning and the end, and this one bacterium, Enterobacter cloacae, had almost disappeared by the end of his weight loss scheme, right? And he took that bacteria, cultured it up, and he stuck it into mice. Mice with this bacterium, this one species in it, put on significantly more weight than mice without it. Almost 75% more weight for the same calorific intake. And that was the only experimental variant between those mice. So you can take one bacterium and put it in a mouse, and the mouse gets fat. And that's fundamental to our understanding of the system. But the opposite is also true. It's a great study from Ruth Lay at Cornell. She looked at monozygotic twins and dizygotic twins. One thing she found was that the human genome actually has some influence. It's limited, but some influence on the bacterial communities that can grow inside us, okay? So if you're a monozygotic twin, your microbial communities between those two twins were slightly more similar, significantly so, but slightly more similar than dizygotic twins or normal siblings. One thing I love about this study, though, is she looked at twin pairs, monozygotic twin pairs, where one twin was obese and one twin was thin. It's really hard to predict obesity from the human genome. Really, really hard. And usually you're only about 50% accurate in predicting obesity when you have a human genome signature, okay? And that varies. For some conditions, it's a lot better. But for general diet-induced obesity, it's really poor. And you've got two twins with an identical genome, and one could become obese and one could stay lean. And she looked at the types of microbes present in those two twins, okay? In the lean twin, there was one bacterium, this Christianicella minuta, that was really abundant in the lean twin and virtually absent in all the obese twins. And she took this and tried to do experiments with it. If we take the microbiome in the stool, so-called fecal microbiota, the bacterial community in the stool, from an obese twin, and we give it to a mouse, just the same as in Liping Zhao's study, that mouse gets fat, right? It puts on weight for the same calorific intake as a normal mouse. If we take that same stool from the same obese person and add this one bacterium, Christianicella minuta, the mouse does not get fat. So this has a protective influence. Imagine if you could take this bug and sell it in Whole Foods for like $5,000 for like a gram. You'd become extremely rich extremely quickly. But we're not unethical. So we have to go through all of the regulatory procedures and try and figure out how this bacterium does this and if there are any nasty side effects. Obviously, it's living in a healthy person, so we don't think it has negative side effects. We also can't just run off and do clinical trials like that. Understanding the metabolic interaction and computationally modeling the metabolic interaction of this organism with the rest of the microbiome and the human is kind of fundamental to pushing forward our understanding of how this bacteria manipulates the body to affect weight change. The work we've been doing here at Chicago with Eugene Chang and Vanessa Leone has started to tease apart the interaction matrix between the bacteria and the host, okay? If you, nice background, right? If you do a night shift or you are chronically jet lagged like I am, you're more likely to put on more weight for the same calorific intake than someone who's not chronically jet lagged or working a day shift, okay? So disrupting your circadian rhythm, the day-night, your perception of the day-night cycle can have significant impacts upon how much weight you generate. And this is because it disturbs the circadian rhythms inside your brain, which affects circadian rhythms in your liver, which alters what types of hormones are degraded and affects the type of energy metabolism that goes on inside our body. Essentially, more energy gets pushed towards adipose tissue, fat tissue generation, than towards respiration. What we saw when we disrupted the microbiome in a mouse by feeding it too much milk fat, so too much animal fat, was that the microbiome's circadian rhythm inside the gut was disrupted. The mice were living a normal day-night cycle. Their brains were ticking away as per usual, but the microbiome's circadian rhythm was disrupted by the over addition of saturated fat compounds, right? And what we saw is that affected the circadian rhythms of the liver, not of the brain, but just of the liver. And when the circadian rhythms of the liver were affected, we saw exactly the same effect as if the circadian rhythms in the brain were affected, in that the mice were put on more weight because the wrong types of hormones were being degraded and more energy was getting pushed towards fat tissue generation. So in this instance, diet-induced obesity is triggering the same metabolic response inside the body that you'd expect from somebody who's chronically jet-lagged or suffering from working a night shift. Yeah?
Why they become obese? Why they don't become thinner?
Well, in this instance, you're actually, because it's pushing the metabolism towards a different route. Now, why is it push the metabolism towards that different route? I don't know. Go ask evolution. But it's interesting to note that this is a repeatable event.
Just a clarifying question with the previous mechanism you were saying, the presence of particular bacterium leads to weight gain, even though the diet is the same for both people. So what's the mechanism there?
Well, we think it's exactly the same mechanism as this.
So the presence of that bacterium either does or does not disrupt circadian.
Potentially, yeah. Nobody had looked at circadian rhythms before we did this study for this kind of process. So in essence, this is a potential mechanism behind which that microbe is affecting change in the human system. Okay.
When the diet is the same.
Yeah. But we don't know what influence Christianicella minuta has in that. Maybe Christianicella minuta influences circadian rhythm of the rest of the microbiome. It could be what we call an ecosystem engineer. It has an impact upon that ecosystem turnover, the ecosystem phenotype, affecting the host back to a normal circadian rhythm. And this may not be the only option. It's maybe not the only study, but it's definitely one that we find particularly interesting and has real causative impact upon the health of the host. So we're interested in where those microbes come from. This is pure ecology. Just the same as if a volcanic island suddenly erupted from the ocean and then plants and animals start colonizing it. And when a baby is born from the amniotic sac, as soon as that amniotic sac ruptures, microbes start colonizing this new island that suddenly occurred, right? The first colonization comes from the mother's vagina if you're born by a vaginal delivery. If not, it comes from the skin if you're born by a cesarean section. So that automatically is your starting culture. And that starting culture kickstarts your microbiome. That's not the only story. When you start breastfeeding, if you start breastfeeding, your microbiome is enhanced by a symbiotic. This is the good prebiotics that are in milk, that's the fats, the lipids, the carbohydrates that mother generates, but also a probiotic. Milk itself, breast milk, has its own microbiome embedded in it. The mother actively recruits bacteria from her gut, from her oral cavity, in through the lymph system and deposits them in the breast duct to form a biofilm, which then actively is pumped into the child, right? So this is another step in the evolution's taken to make sure that we have an appropriate microbiome and an appropriate community to start degrading the right kinds of foodstuffs in our stomach to help us grow up and be healthy, but also to train our immune systems. This isn't actually the only component, though. There's a great study that we're working on right now with Carol over in human genetics, where she has a really interesting component, right? She has studying the Amish and the Hutterites. And in the Amish population, they all live on individual family farms, and the mother and the children and the father, they all muck in the barn, they all work with the animals, and they get all dusty and what have you. But they live a very similar lifestyle, technologically free, to the Hutterites. But the Hutterites live on these big communal farms, lots of families buy a huge tract of land, and they have very mechanized farm areas. And only boys over the age of 14 are allowed in the farm, okay? So the women and the children don't really get that exposure. What's interesting is all of these are vaginally delivered for the most part, unless there's an emergency. They're all breastfed, even if it's by a wet nurse. So they're getting that good starting culture. But the Hutterites have elevated levels of asthma over the US population average, and the Amish have no asthma. So that environmental exposure appears to be also driving the acquisition or the trend towards specific allergic phenotypes. So we're very interested, I don't know if that will work. It won't. We're very interested in where, it did. This bad. We're very interested in where those microbiomes come from. We think that, that a major component of the development of an environmental microbiome, environmentally acquired microbiome, is based on our environmental exposure, where we live while we're growing up. And for the first 2 to 3 years of your life, your microbiome is in high volatility, and it's really accepting anything it can get hold of. This is probably a reason why evolution's driven kids to stick whatever they find when they're picking up in their mouths. We say it's because they're trying to taste it. But no offense to my psychologically relevant colleagues, it also probably has a fitness regimen, right? It causes an evolutionary fitness event. And if they're training their immune system by throwing different types of microbial exposures into their body, it probably has real relevance for their ongoing health. So the microbiome of our environment that we grow up in is highly relevant. And in this Western world, especially in Chicago, where it's either too cold or too hot, we spend 90% of our lives living indoors. As an infant under the age of 1, it's 99%. So this is the ecosystem which we are now readily exposed to. So we did a study of homes where most infants spend their lives to try and understand the dynamic interaction between their developing microbiome and the microbiome of that environment. And we wanted to understand how that microbiome shaped the system, right? So we looked at different families, and the first thing we found was that the microbiome of your home is entirely predicated on which family lives in that home. So this exposure, this shedding of bacterial communities into the space in which that family lives is fundamentally changing the microbiome. Right now, imagine if you grow up as an infant on the Serengeti and you're exposed to raw meats, you're exposed to bush plants, you're exposed to dirt, you're exposed to different animals, you're living in the outdoors and fresh air for the most part, compared to an infant growing up in this environment where the HVAC system is sucking out as many of these dangerous pollutants and pollens as possible, because we're all deadly allergic to everything that goes on in the air. And we want to make sure the house is very clean, so we buy all of our cleaning products and we sterilize everything as much as possible. And we live in an environment which is highly reduced for our exposure. We're getting such a limited exposure to the microbial world. Most of it's coming from our human occupants. And that's fundamentally changing, potentially, the microbial exposure we have. Interestingly, we can use this for forensic applications. In this component, we were looking at a young couple living with a lodger, and we could tell that that young couple was physically interacting in that space because they share more microbes than they did with the lodger. But the lodger was still living in the same airborne environment, in the same house. So his microbiome was still more similar to the couple than he was to anyone else in the study. So we're working with the National Institutes of Justice to determine the forensic applications of these kinds of signatures. And we can actually tell quite a large number of things about who you are based on your microbiome. If I swabbed each of the chairs in this room afterwards, I could tell if it was a man or a woman sitting on that chair. There's a reason for that. These are like a loose fishnet of massive gaps for bacteria, and whatever's in here is being shed onto that chair right now. You can't do anything. This is no barrier function. Trust me. And that's very important for our understanding of our signature impact upon this environment, because I can tell a lot of things about the microbiome, and so maybe we could use that to track who was in a space when they were last there.
So is there any, are there any studies on how long that
72 hours.
To what some portion of
So as long as I get it within 72 hours, I can accurately detect which person it came from.
But I mean, just like if there's a physical interaction between two people, let's say it's a forensic case.
An attack or a rape or something. Yeah, so we can accurately. So if the person's dead, so it's a murder, and we have cadavers that we're working with in Hawaii where we're trying to understand that. The change is actually slowed down because the person's own microbiome is undergoing a significant shift due to the decomposition event. But the signature left on the skin is actually reasonably stable because a lot of those bacteria die and become stable on the surface of the skin. So we think it's about 72 to 96 hours on skin. It's about 72 hours or maybe 36 to 72 hours, depending upon the surface environment in a home. So we have a window in which we can grab a signature. But we've kind of veered away from sexual attacks because there's obviously very good evidence about, because that was a very physical event. And we're more interested in the non-causal, non-physical interaction events that go between two people when they're in a space, because those are harder to track, harder to forensically identify. We can track and map the highways of interaction in this space. So using Bayesian statistics, we derive interaction matrices between people's hands and people's feet and their noses, for example, and the microbes that move between those over time, and the floors and the hand interaction surfaces of the space in which people live. This is actually my house. But if we add dogs into that matrix, dogs actually actively increase the microbial sharing in that space. So they run in and they hug you, and then they go and hug your partner, and then they go hug the children, and they lick everything, and they go outside and dig for a bone or eat a bird and bring all that microbes back in. So they fundamentally alter the matrix and dynamic volume of interaction in that space. So we think they're really good. I actually bought a dog, or rescued a dog from Kentucky on the basis of that study. And it's actually really important because we've actually shown that bacteria associated with dogs can cure or can prevent the likelihood of developing an allergic response in a mouse model for asthma. So this mouse congenitally develops asthma unless you add in lactobacillus from dog feces. So we're paranoid about dog feces, but maybe we should be using that as a new probiotic. But I probably shouldn't say that out loud. But the idea that dogs can, or animals and animal interaction can have a fundamental impact harks back to the Amish. The Amish children are fundamentally interacting with all those animals in the barn, and they might actually be providing a protective event for the development of these atopic and allergenic disease states. So we're working on trying to understand all this inside a new hospital environment. So the Center for Care and Discovery down here on campus, we got in there for 2 months before it was open and operational, and we surveyed the entire space, and then we kept on serving it every day for 10 months, developing over 12,000 samples, looking at bacteria and fungi, diversity in the patients, the staff, the air, the water, and the surfaces. And we have over 8.4 million data points collected about the physical and chemical environment of that space, like carbon dioxide content, the temperature, humidity, etc. And then the patient records tell us everything about what those patients underwent. So we can track increases in microbial diversity, and we can see what impact that has. As soon as the hospital became operational, the diversity shot up. All these human beings moved into that space, and they started shedding that microbiome that we saw in the home environment. And this is all the particles, the bacterial and dust particles that people release when they move into a space that you're all releasing right now. You're sharing microbes because you're shedding microbes into this space. In fact, you're all becoming about 0.00001% more microbiologically similar within the hour you'll spend in this room. And we can see that it mostly becomes human. What we see is this is the black and the red is before the hospital was operational, and then the blue and the green are when the hospital became operational, and it became more human-centric. So the microbial communities there were far more, had an origin from the human population that moved into that space. And so we're using a lot of this information to really understand how microbes interact in the human system. We know that hospital-acquired infections, which was one of the reasons that we got into the hospital environment in the first place, may actually be predicated on commensal organisms, so bacteria that are friendly to us, that we bring into the hospital. And how we're treated in that hospital may turn those bacteria against us. So in this instance, we have gastrointestinal surgery. If the gastrointestinal surgery goes well, we see a normal flora, everything's balanced. But in certain instances, the gastrointestinal surgery, where we stitch up the gut, goes terribly wrong. And there's a reason for this. You hit these things with antibiotics, with radiotherapy. The microbial community's gone a little bit chaos. It's like you know, oppressing the masses inside a city, right? And then you cut open the gut and expose them to oxygen. It's like pumping them full of toxic gas and like, you know, tear gassing the crowds. And then you stitch up the gut and you start sucking out phosphorus from the inside to repair the lining of the cell wall of the body. And then you get nutrient depletion. You've basically taken away their food stuff. And then the bacteria that are left riot. And they don't mean to riot, but what they're doing under a phosphate-depleted environment, under a nutrient-depleted environment, is turning on phosphate and nutrient acquisition strategies inside their genome, which means they co-locate to the gastrointestinal wall and start degrading it. And then you get bleh, the whole system breaks open and the patient dies. And so we're trying to understand how to mediate that. We're developing new strategies. So we isolated all these really dangerous bugs from the ICU here, Enterococcus, Staphylococcus, Enterobacteriaceae, and a Candida fungus. And we started putting them in worms. And every time we put these bugs in worms, it kills the worms. Weirdly, unless we put the Candida and the Enterobacteriaceae together, Enterococcus together, Candida and Enterococcus together cancel each other out. They actually suppress each other's virulence. Either one kills the worm. Together, no death at all. Very strange. But we developed a novel strategy to stop the bacteria from rioting. We developed with the group at Argonne, we developed a polyethylene glycol that's got loads of phosphate molecules in it. We add that back into the gut before we stitch it up, and the bacteria do not become virulent. So our level of survival significantly increases when we have PIPEG, this phosphate polyethylene glycol substance in there. So I want to put these microbes in your record so I can check what your microbiome is doing on a regular basis. To do that, we're developing new sensors. Imagine sitting on the toilet in the morning doing whatever it is you do. I check my emails, you know, probably a bit too much information. Normally I look at my Fitbit to see how many steps I have to do today. Or what if I could look at what my microbiome is doing today and develop an algorithm to tell me what kind of food I should have for breakfast in order to rebalance it to make sure it hasn't gone too far off the tracks. So, you know, similar, this is a kind of profile we might develop from that kind of system. This is a group out at MIT who track their microbiome every day for a year. This is one man, this is another guy. You can see they're microbiologically different. So he has more of the purple bacteria, the Firmicutes. He has more of the blue bacteria, the Bacteroidetes. This guy also has Actinobacteria and Tedocutes. So they have a unique signature. That's the forensic angle. But we can also tell over the year, most of it's pretty stable. The size is due to the density of cells per gram of feces, right? This time, though, he got a salmonella infection from eating a dodgy burrito, and his doctor told him he had norovirus. So he went home and didn't take antibiotics. He got better in the end of the day, went back to the doctor and said, no, I have salmonella. Yabu sucks. My technology is better than yours. in this system, this kid went and lived with his new wife in Bangkok, Thailand. For that 6.5-week period, his microbiome significantly altered when he was living there. He wasn't sick, didn't get a pathogen infection. He was just living in a different environment, drinking different water, eating different food, and it fundamentally altered the baseline of his microbiome, right? So where you are affects your decision-making process. This is very important for precision medicine, for understanding how we treat people. It's not just, oh, I'm going to give you 5 minutes of consultation and I'm going to understand who you are and everything about you. You're almost going back to what the Native Americans used to do, and the fact they used to go and live with you for 6 weeks. The shaman would come and live with you for 6 or 7 weeks and your entire family, and would start to understand your lifestyle. I want to get data that can help me to understand each one of your lifestyles in detail. And that might sound invasive, but it could possibly save your life and actually translate you to having a longer, healthier, better well-being existence. I can't stop there. Thank you very much.